Hi there, and welcome to this induction A-level biology lesson. This lesson is here to give you a kind of taste of what's in store for you if you do A-level biology at Cooper's. So we're going to run through the structure of biology at Cooper's, so how many lessons you have, the different teachers, and then we'll also go through a taste of some of the content that you'll be introduced with at the start of year 12. So to begin with, the example we do is OCR A biology. Okay, so that is our exam board. And these are the three A-level teachers that will be teaching you, or potentially teaching you next year. You've got Miss Foster, who is the head of biology. You've got Mr. Chiobanu, and then you've got myself, Mr. Meads. All right, so you'll have two of us in whichever combination. So you could have Miss Foster and myself, you could have Miss Foster and Mr. Chiobanu, or you could have Mr. Chiobanu and me. All right, and these are our codes. So anytime you need to email us for whatever reason, it will be cfo@cooperscobon.co.uk. Okay, if you wanted to contact Miss Foster, ACI for Mr. Chiobanu, and then IME for myself. So these are your three A-level biology teachers that you potentially could have in the start of year 12. So just a bit of a breakdown of the course structure. They've gotten rid of coursework, as you should know from your GCSE. So we have to do 12 assessed practicals over the course of the 2 years. You'll be assessed on how practically competent you are, basically. And at the end of the year, it's either pass or fail for your practical endorsement. Your practical, though, does not affect what grade you get at the end. So your grade that you get from these three papers is separate to that practical endorsement, which is a pass or fail. However, if you are looking to do any scientific courses at university, they will usually ask for a practical endorsement. So it still can be very important for you to get that. So you get those 12 assessed practicals over the 2 years, all your exams at the end of year 13. So you have your terminal exams at the end of year 13. You have 3 papers. You have 2 lots of 2 hour 50-minute papers, and then one hour 30-minute paper. In terms of in class, you'll have 5 lessons per fortnight with each teacher. And because you've got 2 teachers, that's a total of 10 lessons of biology per fortnight. So 5 of each teachers, and we'll teach different sides of the course. So it won't be both teachers teaching the same content. We'll have our own content to teach you. You'll also have an assessment period once per fortnight, okay, which will not necessarily be a biology teacher, but that's time for you to catch up on any work or any assessments. And then a bit of information about the transition tasks. So hopefully you've seen, or potentially will be seeing, these transition tasks. These are to help you make that jump from GCSE to A-level, to help you understand the level we're expecting you to work at in terms of the detail of work, in terms of just the sheer amount of work you're probably going to have to work yourself, put yourself through. And just to give you an idea of what we are looking for. R1 consists of 4 sections. So these first 2 sections, cells and biological molecules, we do these because these will be the first topics you do with each teacher. So with one teacher, you do cells and microscopes, and then with the other teacher, you'll do biological molecules. So these are foundational stuff in biology, as you should know. So that's why we've got these 2. And then some basic skills you need to have as an A-level biologist, math skills, which are so important in A-level biology, and scientific and investigative skills. So knowing key terms and what key terms mean in relation to the methodology, the scientific method. So that's also important. So you've got these 4 sections to complete as your transition task. These should be complete by the time you arrive in September. So we can have a look at them and see how you lot have got on. So what we're going to be looking at today, and to give you a little bit of a taste, so this is what you would have done during your induction day. Unfortunately, obviously, because of the certain circumstances, we can't do that. So I've tried to adapt it as best as possible to the circumstances. We're going to have a quick peek at biological molecules. So this can give you an idea of the level you need to kind of start working at and the level of detail we expect from you as A-level biologists. So you probably will need a pen and paper if you want to do some writing down. There are going to be questions. So when I give you a question such as this, you can pause the video and then write down your answers, and then I'll go through it as well, just like a normal-ish lesson, just I'm talking at you through a screen. So first things, when we look at living things, obviously biology, the study of living things, roughly 99% of living things are made up of just these 4 elements. So I'd like you to write down what you think those 4 elements are. So if you want to pause the video and give it a go, off you go. Okay, so let's see how many you got right. So carbon, hopefully we should know we're carbon-based organisms. All life on this planet is carbon-based. So that's obviously going to be a big one. We've got oxygen, which is another important one, hydrogen, and then finally nitrogen. So these are the main atoms for the building blocks of life. How many do you get of those? Did you manage to get all 4? Did you miss out some? Now, these building blocks are used to make some biological molecules. We're going to have a talk through some of these. So, what biological molecules are made from these elements? So, what's just made out of hydrogen and oxygen? What's just made out of carbon, hydrogen, oxygen? What's also made out of carbon, hydrogen, oxygen? And then what's made out of carbon, hydrogen, oxygen, and nitrogen? Now, if you think about biological molecules, the best way to think about it really is actually thinking back to maybe key stage 3 when you talked about different food groups. So that's my little hint for you. See if you can work it out from food groups. So if you want to pause the video and see if you can work out what biological molecules are made from these elements. Off you go. Okay, so hydrogen and oxygen, we should probably work out, is water. So we'll spend a lesson or two on looking at water and why it's so important for living things. Next ones, we've got carbohydrates. So carbon, hydrogen, and then the oxygen in there. CHNO, the other one, is our lipids. And then the final one at the bottom are proteins and nucleic acids. So nucleic acids, stuff like your DNA and your RNA. The ones we're going to touch upon today, just in this very short induction lesson, are going to be carbohydrates, lipids, and proteins. So these are the main 3 ones we're going to be looking at today. But in the real thing, obviously, we'll spend time looking at water, and we'll spend a whole chapter looking at proteins and nucleic acid together. So here we have a table looking at the name of the biological molecule, its monomers, and the polymers. So a monomer, mono meaning one, so it's the one singular unit that makes up that biological molecule. So for example, carbohydrates, the monomer of a carbohydrate is called a monosaccharide. And then there's different examples of monosaccharides, which we'll talk about. Then what I want you to do, or now what I want you to do, is have a look and see if you can work out what the ones that are missing are. So what is the biological molecule that is made up of amino acids as its monomers and is called a polypeptide when it's made up of multiple monomers, which is a polymer? And then nucleic acid, we've got polynucleotides. What's the monomer for that? So if you just want to pause it, you can draw out this table if you want, or you could just work out what the missing bits are. Pause it, off you go. All right, so let's have a look. So carbohydrates, the monomer of a carbohydrate is a monosaccharide. So the polymer is a polysaccharide. So when you think of things like starch, okay, or glycogen, that is a polysaccharide because it's made up of multiple or many monosaccharides. What is the biological molecule that's monomers are amino acids and that the polymers are called polypeptides? Well, that's going to be our proteins. So our proteins, we've got an amino acid, and the polymer is a polypeptide. So polypeptide chain or multiple polypeptide chains can make up a protein. And then the last one, nucleic acid. What is a monomer of nucleic acid? Well, it's a nucleotide. So hopefully you know from your GCSE that a nucleotide is made up of that phosphate, a sugar, which can be ribose or deoxyribose sugar, and a nitrogenous containing base. So these are just the terms, some of the terms you're going to have to learn when looking at this. So let's break it down through our first one, which is going to be carbohydrates. So when we look at carbohydrate, we can see here what this looks like. So we've got a monomer, which is a singular, which can be a monosaccharide. We've got a dimer, which in this case for carbohydrates is a disaccharide, and that's when 2 monomers or monosaccharides are chemically joined together to make a disaccharide. And then a polymer is multiple monomers chemically joined together. So in this case, what we have here, this monomer or monosaccharide is glucose. We've joined here 2 glucose together, which gives us maltose. And then we have multiple glucose molecules, which makes up a type of starch, which we'll talk about. So here are some of our monosaccharides. The ones you really need to know are going to be your hexose sugars, which are made up of 6 carbons, which are your glucose, fructose, and galactose, which you'll learn about when we look at carbohydrates. Your pentose, which are your ribose and deoxyribose, you'll need to know those when we look at nucleic acids. Triers, you do not really need to worry about, but there are such things as tri sugars, such as glyceraldehyde. So disaccharides. Now, we've got several here that you need to know. So you need to know what makes up these different disaccharides. So remember, disaccharides are two monosaccharides put chemically joined together. They don't need to be the same monosaccharide. So sucrose is made up of a glucose and a fructose monosaccharide chemically joined together. Maltose is glucose and glucose. And our last one, lactose, is made up of glucose and galactose. So all of these we know are monosaccharides, and when we join them together, these are our disaccharides. Now, there's some ways in which we can test for these different carbohydrates. We have iodine to test for starch, or iodine solution to test for starch, where we should see if it's a positive, go from yellow to blue-black. And that is a key term you need to know. Blue-black is the actual scientific term for it. And then we've got sugars. So we've got two types. We've got reducing sugars. We use Benedict's solution for this, and we should see it go from blue to brick red. And then we've got non-reducing sugars, which we need to do a step before we do the Benedict solution, where we add hydrochloric acid to break them down into the two singular monosaccharides. Now, the reducing sugars, if we go back, are all of our monosaccharides. So all monosaccharides here, especially our hexose sugars, are reducing sugars. Maltose and lactose are also reducing sugars. The only non-reducing sugar you need to worry about is sucrose. So reducing sugars, if it's got glucose, galactose, fructose in, this test will test positive, go from blue to brick red. If it is maltose and lactose, they are reducing, so they'll also go from blue to brick red. Sucrose during this test would stay blue, but then if we did this test, it will turn brick red. Because what we do by using hydrochloric acid is we break down the sucrose into fructose and glucose, which are reducing sugars, which would then turn it brick red. So just to show you, obviously we would do some of these in school. So part of the induction lesson would have been doing practical, but instead we're going to have to show you via a kind of video. So we're going to look at doing our iodine test and how we do our iodine test. Some of you probably all done this before. So we can detect starch by using iodine solution, the iodine test. So first what we do with this is we add a few drops of iodine solution, which would be that yellowish orange brown color, as you can see here. We add it into our samples. You can do this also in a drop-in tile. So we add it in, and we can see here from this, we'd be able to tell which one is got starch in, the one that's gone blue-black. And the reason why it goes blue-black, and this is a little bit extra, is because it makes a complex. So the iodine binds in between the starch. So this is our starch molecule, which you can see here is curled. It binds there to make a complex, polyiodide starch complex, and that's what turns it that blue-black color. If there's no starch, there's nothing able to make that complex. It doesn't go that blue-black color. So that's the iodine test. Now we're moving into our proteins. We'll look at the other tests in a little bit later. So with our proteins, this is an amino acid. Now, when we learn about amino acids, what you'll know is there's a basic shape, which includes this amine group, this carboxyl group. We've got this then hydrogen, and now this group here, this is CH3, which is the amino acid alanine, but that is a variable group. So that group can change based on what type of amino acid it is. So there's 20 amino acids, so that has 20 different things that attach onto this carbon here. Right? When we join two amino acids together, chemically join them, we get a dipeptide. So here we've got glycine and alanine, which are two amino acids chemically joined together. Now, when we keep joining proteins up, we get a polypeptide. Now, polypeptides are interesting because it doesn't just stop at them joining up. What happens is you have different folding that takes place, and this different folding leads to different levels of structure, which you can see on this side. So with protein, what we can see here is the primary structure is just purely the amino acids that make up that structure, as we can see here. So it's just the different amino acids that are joined together. The secondary structure is how it initially starts to fold. So it's got two ways it can fold, which are the alpha helix, which you can see here turns into a helix, or beta pleated sheets, where it kind of folds in on itself. So that's the secondary structure where we have these folds. The tertiary structure is where it gets its final kind of three-dimensional shape. This happens with more folding, so it folds even more in itself, and this is where a lot of bonding starts to take place, different types of bonding. We've got ionic, we've got disulfide bridges, we've got hydrogen bonds. All these things turn it into that 3D shape. Now, some proteins stop there. Other proteins, such as hemoglobin, are made up of multiple of these polypeptide chains. So then these polypeptide chains will join together to make the quaternary structure. So the quaternary structure is multiple polypeptide chains that have now joined together. So with hemoglobin, which I think this is actually showing, you have 4 different polypeptide chains that join together to make it up. Now, when we want to test for protein, we can use the biuret solution. I'll show you what the biuret solution does a little bit later on, but it's the mixture of sodium hydroxide and copper sulfate solution. And what we see is a change from blue to lilac color, so purple. Our last ones now are lipids. Now, lipids aren't polymers. So a polymer is multiple repeating units that are the same or close to being the same. So with proteins, they're polymers because they're made up of amino acids. Carbohydrates, because they're made up of monosaccharides. Lipids, on the other hand, aren't actually polymers because they're not repeating units of the same thing. But because they are big molecules, we call them macromolecules. And there are 3 main types you're going to come across when we talk about lipids in A-level biology. And these are our 3 ones here that we can see here. So we've got this one type here, we've got this type here, and then we have this type here. So this one, which is very common, which is the one you will know probably, and this is the one kind of that can make up food we eat, are called triglycerides. So these make up the fats and oils we eat. Triglycerides, you've got a glycerol molecule here with 3 fatty acid chains. That's where we get our tri from. We've got 3 fatty acid chains that come off it. And depending on these fatty acid chains, depends on whether we call it an unsaturated or a saturated fat. Now, over here, we've got one that's very important. Some of you might have heard before, but it's very important to do with our membranes, especially our cell surface membranes. These are called phospholipids. So they have a phosphate head here with 2 fatty acid tails coming off. They also do have a glycerol molecule, which you see here. So they've got a glycerol molecule like triglycerides. They've got 2 fatty acid chains. And then on this final one, they'll have a phosphate head, which we can see here. And then finally, our last one is this one here, which an example of it is cholesterol. These are steroids. So steroids are also types of lipids. An example of that is cholesterol. So those are our lipids. Now, to test for lipids, we can do the emulsion test. This is where we mix ethanol and then add water and shake. And we should see, turns milky, it forms what we call a white emulsion. So let's have a look at how this happens. So we've got our emulsion test here. What we need to do first is, once this loads up, is we add ethanol, which you can see here, to our sample. Now, fats are non-polar, which means they haven't got a charge to them. And the only thing that can dissolve non-polar molecules is other non-polar things. So alcohol is also non-polar, as it tells you here, so it can dissolve. Water is polar, so that's why it can't dissolve fats. That's why we've got to use ethanol. So then we dissolve it in there. And then what we need to add to this is water, because this will now create that emulsion because it can't dissolve in water. So we shake that, and what we see here and up here form, as it's going to show you now, is the white emulsion. So that shows us that there are lipids present, all right? Because lipids aren't soluble in water, all right? Because water is polar, lipids are non-polar. So that's what we do for our fats, our tests, our lipids. We would use the emulsion test. Now, this is what you would do if we were in our induction lesson. So I'm going to take you through it anyway. You would be testing urine for 2 biological molecules. We'd be testing it for reducing sugar, and we would be testing it for protein. So I said we're going to go through these tests, so that's what we're going to do now. But I want you to have a think. While testing the urine, which we can do whilst you're just thinking about, or what I'm teaching you about those 2 tests, have a think about what diseases might give a positive test for sugar in the urine and protein in the urine. So have a think, what will give us a positive result? What disease would cause there to be a positive result for sugar in urine? And what disease or thing could cause issues that lead to protein in the urine? Just to help you understand, there should never be any sugar or any protein in someone's urine. So there shouldn't be any of that. It should be reabsorbed back into the body. So let's have a look at the testing first, and we can come back to this answer. So the two tests you'd be doing would be your Benedict's test and your biurate test. So let's have a look at the first one, which is our Benedict's test for testing reducing sugars. So monosaccharides and some disaccharides are reducing. We talked about the disaccharides was our maltose and our lactose are reducing sugars. So what we do, if you had your different samples here, we would add equal amounts of Benedict's solution to each test tube, each sample. You can see here. Sorry, it looks a bit weird with it stopping. So we've added Benedict's solution into each one. It has copper in there, ions that give it that blue color. What we then would have to do, and what you would have to do, is you'd have to stick it in boiling water for roughly 5 minutes. What's going to happen then, if there is a reducing sugar present, you will see it form a precipitate. Right? If it has high amounts of sugar, it would form a brick red precipitate, which we can see here. Okay? Now, what we can see, as you'll show you, is depending on the relative amount of sugar, gives us different colors. So it actually gives us a gradient, which you can see here. Blue means there's no reducing sugar, and as we get more towards brick red, we can see then that there's reducing sugar present. Now, if we wanted to test for sucrose, which is a non-reducing sugar, we would have to use our hydrochloric acid to break down the sucrose first. So this is what we do to test for non-reducing sugars. We heat it, we boil it using hydrochloric acid. So we're boiling it, it's got the hydrochloric acid in the sucrose sample. What we then need to do is to make sure we neutralize it. If we do not neutralize it, the Benedict's test will not work. So we use sodium hydrogen carbonate to neutralize this acid in there, which we can do here. So we can neutralize it with the sodium hydrogen carbonate, which is going to happen in a second. There we go. Then we can do the Benedict's test. And once again, it's the same as last time. You add Benedict's solution in, you then heat it up again. And now what we should see, if there's a non-reducing sugar present, we should see that it got a negative test on the first time we did this. And now we've done it the second time using the non-reducing sugar test, it should come back positive. Which you can see here now, because it's turning that brick red color. Now, when we talk about it, like with iodine, is blue-black for positive. A brick red is the correct terminology we need to use for high sugar content. Now, let's have a think. At the start, we asked what disease potentially could lead to getting a positive result for testing urine for reducing sugar. Well, what it would be diabetes. Okay? So one way in which the body can try and level the amount of sugar. So think diabetics, they can't control their blood sugar level. If they eat something, they're not producing insulin to take it into the liver and the muscles. So one way in which your body can try and ensure that blood glucose levels do not get too high, which can be very dangerous, is to get rid of it by urine. And so what they would do is they could test the urine for sugar. If it's got sugar in there, that gives us a good idea that potentially they have got diabetes. A way in which doctors used to test for this is they would actually taste the urine, because if it's got sugar in it, the urine's going to taste sweet, and that's how you can diagnose diabetes. Obviously, now we don't do that. We've got other methods to do it, as one of the ones we're showing you here. So that is how we do the Benedict's test. So that's what you would have done if you're in the lesson, if it wasn't for the certain circumstances we're in. Finally, we can look at the biurate test now. So the biurate test is testing for protein. So let's have a look at what happens here. So what we do is we add a few drops of biurate reagent, which is made up of copper sulfate and sodium hydroxide. We add it to our test tubes, and we can shake it just to mix the sample with the biurate reagent, as you can see there. You might already be seeing something taking place. You don't need to heat this or anything. Now, the solution will turn purple or violet color in the presence of peptide bonds. So the peptide bonds are the bonds that join amino acids together. If it's got a peptide bond, it's going to change that color. And as you can see here, once again, this is where it joins with those peptide bonds to give us that lilac color. The more bonds you have, so the more protein you have, the more intense the color change. So the more lilac it goes. Now, disease-wise, once again, you shouldn't have proteins in your urine because proteins shouldn't be able to fit through what we call the fenestrations in the kidney that allow the filtering to take place. They're too big to fit through the holes, basically, where everything is filtered. So if you've got protein in your urine, that usually means that either you've had some form of kidney infection where it's attacked that lining so the proteins can get through, or hypertension, so high blood pressure, because literally the pressure is so great it's physically forced those proteins through, and that's how it ends up in your urine. So those are some of the diseases potentially you could be diagnosed from having protein in your urine. And that is it. So I hope you've had a good taste of what's to come with A-level biology. I know it's a little bit harder because we're not actually in the lesson, and it'd be much easier to understand if we were doing it. But I hope it's given you some idea of what's in store, and I hope you've actually enjoyed it and looking forward to what potentially can be happening in September. The transition tasks, as I said at the start, are there to also help you with getting an idea of what's to come in September. So I hope you enjoyed it. Goodbye.